Hello and welcome to my talk. I'm Ruoshi Zhang and I'm a PhD student at the Max Planck Institute for Biophysical Chemistry. Today I'm presenting our work on sensitive identification of phages from the CRISPR spacers in their prokaryotic hosts. Our work focuses on resolving the interaction between bacterial phages and their hosts, which are bacteria and archaea. Phage-host interactions are central to the ecology and evolution of microbial communities. Until recent years, the infection relationship of phages and hosts was still primarily confirmed by laboratory cultivation. However, most of the microbes cannot be isolated or cultivated in the lab. Luckily, the advancement of next-generation sequencing allows us to discover new phages and the hosts through culture-independent metagenomics by directly sequencing from different environments. The question who infects whom is crucial to characterize the increasing number of phages and hosts uncovered from the metagenomes. At the moment, most phage sequences are not associated with any host, and likewise, many bacteria and archaea have not yet been associated with any phage. There are many features that help us to link phages to their hosts, some of which do not rely on alignment, such as comparison of k-mer frequencies of phages to the host reference genome, and some are alignment dependent. Our approach uses CRISPR spacers, which record the past infection in the host genome. The CRISPR-Cas systems are adaptive immune systems in bacteria and archaea that protect the organisms against viruses and other parasitic mobile genetic elements. After surviving a phage infection, bacteria place a short DNA fragment of the infecting phage as a spacer into the CRISPR locus. And the spacers are separated by a repeat sequence. The spacer later will be used for targeted cleavage of future invaders' DNA. Therefore, by matching the spacer to the phage genome, we can infer phage-host relationship. It is perhaps important to point out that a CRISPR-Cas system is only possessed by 40% of the known bacteria and 80% of the archaea, and the prevalence is highly environment dependent. So by nature, bacteria that do not harbor a CRISPR-Cas system will be missing in the host spacer database. The state-of-the-art method aligns individual spacer sequences to phage genome using BLASTN with stringent filtering that is allowing only up to 2 mismatches. This method had been widely used in studies since perfect CRISPR spacer matching to the phage leads to successful defense of the host organism, and therefore the prediction made from these matches are very specific. But the method is not very sensitive as it is limited to find very close matches. BLASTN can also become too slow if the database scales up, especially when analyzing large-scale metagenomic datasets. To address these limitations, we developed a tool called SpacePHARER. The core SpacePHARER algorithm starts with 6' translated spacer sequences and phage protein sequences. We perform an all-against-all search using MM6 as it is much faster than BLAST. Then, we calculate a combined score for every phage-host pair with all the significant hits given their p-values, which represents the strength of evidence. This combined score will be a measure for possible relationship between a phage and a host. SpacePHARER improves sensitivity on 3 aspects. First, we perform a translated search because the phage genome is mostly coding, and due to selection pressure, viruses tend to mutate their genome with minimal changes to the amino acid sequences. Nevertheless, we still include the nucleotide level homology by realigning all protein hits again on the nucleotide level. The nucleotide hits with very few mismatches is very specific, so we use these information to prioritize hits. Second, the spacers are very short and even shorter when translated. we optimize search parameters for short and highly similar protein sequences. The multiple spacers matching to the same phage genome is, of course, a strong evidence of phage-host interaction. And this could be a result either of multiple infection events or of a special process observed in certain CRISPR-Cas subtypes called priming, where the system quickly incorporates multiple spacers from the same viral genome. Some of these spacers might not be perfectly matching with the phage, but together they can still contribute to the prediction. We take advantage of these cases by combining the homology signals from all the significant spacer hits. SpacePHARER makes predictions de novo, that is, we do not rely on any previously known phage-host relationship. Instead, we perform a false discovery rate control to select for the true positive matches. The false positive rate, or FDR, is defined by the proportion of false positives among all positives. To do this, we run the SpacePHARER workflow with the spacers against a no-model database where we do not expect to find any hits, for instance, by inverting the phage proteins. Then we estimate the fraction of false prediction by the number of matches we find in the no-model database. We filter for the true positive phage-host pairs with a FDR cutoff of 0.05. Some CRISPR-Cas systems require a very short, highly conserved protospacer adjacent motif, or PAM, flanking the viral target to prevent autoimmunity. In many cases, identifying the correct PAM next to the protospacer region will increase the accuracy of the prediction. For each spacer hit, SpacePHARER takes the flanking sequences up and downstream of the protospacer and search it in the list of consensus PAM patterns and report if a possible PAM is detected. The performance of our tool is assessed by analyzing two public comprehensive spacer and phage datasets. For the benchmark, we use two different no-model databases. We compare the performance with the most commonly used BLASTN settings by other studies. We additionally perform the SpacePHARER FDR control step on the BLASTN search output. The prediction power of both methods is evaluated on the number of true positive predictions at different FDR cutoffs. On the comprehensive spacer versus phage datasets, SpacePHARER reported 4 times more phage-host pairs than BLASTN at the FDR of 0.05. On a desktop PC, SpacePHARER took 12 minutes to process the datasets, while the BLASTN search took 47 times more. If the host taxonomy labels are supplied, the weighted lowest common ancestor, or LCA, of the phages can be automatically computed using the MM6 taxonomy module, which was presented by my colleague Milot earlier. We search a validation dataset which comprised of 800 phages with a well-annotated host taxonomy, and we counted the number of correct versus incorrect host predictions up to every taxonomic rank. We use a stricter FDR cutoff of 0.02 for the matches to be considered for the LCA procedure. The number of predicted phages is lower on the lower ranks due to the correction of the maximal ranks allowed for the LCA by average nucleotide sequence identity, so that we don't predict an overly specific host taxonomic label where there is not enough evidence. Here, SpacePHARER shows a higher precision on host prediction than BLAST on all taxonomic ranks. We provide SpacePHARER as an easy-to-use command line tool, and we also offer a pre-formatted spacer and phage databases for download and with one command. And we also offer an easy predict workflow where users can directly search self-provided spacer files in common formats against phage databases. The output of SpacePHARER consists of phage-host matches and with their spacer hits. The line in blue contains sessions and the combined score of a phage-host pair, and each following line is a spacer hit with information on p-values, aligned region, and the possible PAM up and downstream on both strands. Here, I also want to mention that SpacePHARER is also available for installation on Conda. To summarize, we developed SpacePHARER for fast and sensitive de novo prediction on phage-host relationship. We showed that it is up to 4 times more sensitive than BLASTN, thanks to searching with translated sequences and combining multiple spacer hits. We also showed that SpacePHARER is fast enough to analyze large-scale metagenomic datasets. We hope that SpacePHARER can contribute to a better understanding of the interaction and dynamics between phages and microbial communities. With that, I want to thank all the people who are involved in this project, especially Milot, Ellie, and my supervisor Johannes, and all Zeding Lab members. I also want to thank the funding agencies for support and the conference organizers for giving me the opportunity to present our work. SpacePHARER is published as a manuscript, and for more details on how to install and use the tool, please check out our GitHub repository. Thank you for your attention, and I'm happy to take your questions.